In this video, we're going to take a look at the molecules, primarily proteins, that are involved in defending the body against infection and aiding the other innate host defenses that are available. And the first of these molecules is interferon, or the interferons. There's not just one interferon. So should an animal cell, like a human cell, become infected with a virus, it may produce these molecules that have been named interferons. They're just small proteins that bind to a nearby cell and then interfere with the ability of the virus to replicate in that cell. So when a neighboring cell when it binds that interferon, it produces a class of proteins that are called AVPs, or antiviral proteins. And so these antiviral proteins will interfere with the virus. So interferon is made by a virus-infected cell. That's the primary means, although we'll see in a second that there are other cells that can make interferons. But cell gets infected by a virus, it produces interferons that releases the interferons into the extracellular fluid around it. The neighboring cells then, in response to noticing interferon in the extracellular fluid, produce antiviral proteins, AVPs, and they will limit the ability of the virus to reproduce should that nearby cell become infected. And so the point of interferon then is to limit the spread of the virus through the body. Because if nearby cells can be infected but cannot have their machinery taken over by the virus and have any virus particles made there, then the virus is blocked. So two classes of interferons, type 1, are the ones that are produced by cells that have been infected by a virus. And they include alpha interferon and beta interferon. So these are two groups of interferons that are type 1. Type 2 interferons, they're termed gamma interferons. They're produced by lymphocytes and by natural killer cells. So lymphocytes and NK cells can also make interferons, and they do it only once they've been exposed to the virus's antigens. They don't have to be infected by the virus in order to produce these gamma interferons. So when these were first discovered, and I actually remember this, it was in the late 70s, early 80s, if I remember right. There was a Time magazine article that I read with a lot of interest because it sounded like a really cool way to be able to fight disease. And, we thought, oh gosh, interferons, they're going to be the next big thing. And it turns out that they are not. The promise that they seem to hold has not been met, but they can be used to treat some diseases. So again, they don't cure, but they can treat some cancers, those that have a viral origin. They can treat genital warts, so the genital herpes, and hepatitis C, which is a viral disease, can also be treated with interferon. Okay, so those are the interferons. Let's keep going. And the next of the molecular defenses that we're going to look at are the complement proteins. The complement proteins are this very large family of big plasma proteins. They're made by the liver, they're found in the blood, and they can be up to 20% of the plasma proteins found in blood. And they're named for their ability to complement the action of antibodies. And there have been some that suggest that while antibodies do good, it's really complement that takes out the invaders that make their way into the body and that prevent disease. So what is it that complement can do? Well, three things. It can make it easier for phagocytes to phagocytize whatever material is that's been found in the body that shouldn't be there. It can cause bacteria to lyse, so cut holes in bacterial cells, causing them to die. And it can do that with both bacteria and with the viruses that have a membrane around them. So those enveloped viruses are also subject to complement. And then last of all, it can generate peptides that help to regulate inflammation and other immune responses. So the way that complement works is that it causes what's termed a cascade of chemical reactions. So let's say we have one complement protein and it binds to, so here's a bacteria, here's the antibody, it binds to an antibody. This complement protein now becomes an enzyme, and anytime it stumbles across complement protein, so we'll call this one A, anytime it stumbles across B, it breaks B apart and creates an active enzyme. So now we have B part 2, which is an active enzyme, and every time it finds C and binds to it or interacts with it, then it breaks C apart into active forms. So here's C2, and now C2 acts as an enzyme, and so on. So the thing that makes these cascades so amazing is that this enzyme, say B2 here, it can activate this C protein, and as soon as it does that, then it can activate a second C protein and a third and a fourth and so on. So it creates this cascade where the first enzyme can make a little bit more enzyme, and then each of those can make even more enzymes in the next downward spiral of this. And then each of these is going to make more. And so, as you can see, by the time we get to the end product, then there's a huge number of those end product proteins. So it yields almost exponential production of the proteins that are later in the cascade. And so it's a way of magnifying the effect of the action, because it can make so many workers that will go and cause whatever the effect might be. So hopefully that makes some sense. So let's dive in then and get a little deeper into how complement works. So 3 pathways. There's the classical pathway, called classical because it was the first one that was discovered, in which an antibody binds to a foreign antigen, and it activates the complement proteins C1, C4, and C2, and then that leads to the effects that we see. There's a second pathway called the lectin pathway, and so this time it's macrophages that, as they do phagocytosis, they release cytokines, and this causes the liver to release a molecule called lectin. Lectin binds to antigens on the bacteria and to viral surfaces, and once it binds, then it skips C1, but it activates C4 and then C2. So we got the classical pathway, which is C1, followed by C4, and then C2. The lectin pathway kind of hops in and goes straight to activating C4 and then C2. And then there's the alternative pathway, sometimes called the properdin pathway. In this pathway, antibodies aren't required and lectin isn't required, but instead, the complement proteins factor B, factor D, and factor P, which is properdin, hence the name, these bind directly to the foreign antigen. And once we get there, then we get to activation of the complement pathways. So we have 3 ways to activate complement, but that's where we're trying to get to, is that if these 3 factors bind to the antigen, or if lectin gets made and we make C4 and then C2, or if we do the classical pathway that starts at C1 and then moves to C4 to C2, finally we get to the activation. So once C4 and C2 have been made, or BDMP bind to the antigen, then that triggers the reaction using complement protein C3 through C9. So now we're going to see all the other complement proteins get involved, and that will trigger one of 3 molecular defenses. It will cause opsonization, it will enhance inflammation, or it will produce membrane attack complexes, or MACs. So 3 things that can happen or will happen once we activate the complement system, once we get through those first steps to get complement going. And so we're going to look at those 3 things over the next 3 slides. So don't feel like I'm going to leave you hanging about what opsonization is, because I know you are very eager to find out. So let's take a look. We'll start with opsonization. So opsonins, these are specialized antibodies that will bind to and then coat the surface of an infectious agent. So here we've got an infectious agent, and when these opsonin antibodies coat it, then C1 will bind to C4, to C2, to C3, and then to C5, and opsonization will occur. So we'll come down this pathway with that made. So C3b, which is so if we have C3 here, and it breaks into 2 different components, C3a and C3b, C3b will bind to the microbe surface. Now, if C3b is bound to the microbe's surface, and so here it is, it's bound right there, then there are specialized receptors on phagocytes that can bind to C3b. And now we've adhered our phagocyte onto the bacterium using C3b as the means to connect them together. And once they've adhered, then it makes it easier for phagocytosis to occur. And so this opsonization, it can overcome the anti-phagocytosis effects of the M proteins that are found in bacteria, and we talked about those before, and make something that otherwise would be very difficult or impossible for a phagocyte to consume, to engulf and then break down, make it possible for them to do that. So opsonization is making it possible for the macrophage to be able to do phagocytosis to the bacteria or viral particle or whatever it is. Okay, so that's opsonization. The second is that the complement pathways can trigger inflammation. So once again, we have C1 binding to the antibodies. That then creates C4b as we split up C4, which then is an enzyme that makes C2a as we split up C2, which then acts as an enzyme to split up C3 into that C3b that we've talked about already, and C3a. So if we have C3a around, and then if we have C5a around, and C4a, which is right here, so these 3 together will trigger positive chemotaxis by phagocytes. So we get phagocytes coming out of the blood and into the tissues where those 3 complement proteins are found. They will also bind to basophils and to mast cells, and they cause them to release histamine. So out comes histamine from the basophils and the mast cells, and so these 2 things are going to trigger inflammation and call phagocytes in to come and munch down on whatever the is causing that inflammation, has triggered this complement response. Okay, so opsonization, there's inflammation, and then last of all is the production of these membrane attack complexes. So once again, we got to get C3 built here. So we've made it down. We split up C3 into C3b. C3b then acts as a trigger for a process called immune cytolysis. So here's cyto, that means cell. Here's lysis, that means breaking it up. So this is cell destruction. And so what happens is we get C5b made. So we got C5b. C6 is around, and so is C7. And together this forms this large complex called C5 B, C6, C7. So we get this large proteins all kind of wadded together. And they go and attach themselves and insert themselves into the cell membrane of the microbe. And then C8 comes along and joins in. And when C8 joins into the complex, that then lets C9 and up to 15 of these C9 molecules assemble and produce a membrane attack complex. So here we see a membrane attack complex where these little blue things are showing C9. So C9 makes this ring that pokes a hole through the membrane of the microbe, whatever it is. It could be a virus, it could be a bacterium. And so if it's a bacterium, it dies as the cytoplasm leaks out through these MAC pores that get punched all through the membrane around that microbe. If it's a virus, then that lets in enzymes or whatever that will inactivate the virus. So these immune cytolysis effects of complement are very, very important as well. Now, so why doesn't all the host cells, why don't they get these MAC complexes cut through them so that they break up too? Well, fortunately, the host cell is protected because it has membrane proteins that prevent complement proteins from binding to the membrane. So it won't let the C5b, C6, C7 complex make itself part of the membrane. Okay, so there we've seen it. Opsonization, stimulation of inflammation, and the development of these membrane attack complexes. So once a complement is activated, that reaction cascade occurs rapidly. And just a small, as we were saying before, just a small quantity of the microbial antigen can activate just a few molecules of C1, which then will activate even more of the next C complement protein and more until we have this huge quantity of whatever the final proteins are that cause opsonization, inflammation, or immune cytolysis. And it's possible for people to be born with deficiency, the inability to make these complement proteins, and that can create then in these individuals the inability to resist infections. So that's a tough thing if you've got that happening. Okay, so a third of these molecular defenses is the acute phase response. And that is a response to acute illnesses. So illnesses that come on, as they come on, that causes an increase in acute phase proteins. They're just a class of another one of the plasma proteins that are made by the liver and found in blood plasma. So they're made as the liver is responding to one of the cytokines that macrophages make as they ingest pathogens. So macrophages produce this interleukin 6. Interleukin 6 talks to liver cells and causes them to make the acute phase proteins. And here's what happens. So two of these cytokines, there's the C-reactive protein, or CRP, and it binds to the pathogen, to the phospholipids in the membrane of the pathogen. And the second one that we understand best is the mannose binding protein. Mannose is a sugar, and so it's found in cell membranes. And so MBP, mannose binding protein, binds to the mannose. And once these acute phase proteins are bound to the membrane of the pathogen, then they act just like opsonin does. So this is like another way of triggering opsonization. So it activates complement. We see the proteins binding and causing or allowing phagocytosis to occur more easily. Okay, so to summarize then the entire chapter, we have this one figure. I think this is a good one to really pay some attention to. So innate host defenses include the physical barriers of skin and mucous membranes. They include the cells that do phagocytosis, so the macrophages, both the wandering and the fixed macrophages, neutrophils, eosinophils, and monocytes. Then there are the white blood cells that perform extracellular killing, like the natural killer cells do. There's the inflammatory response and all of the effects that it has of bringing more blood to the area, stimulating phagocytosis, causing localized warming, the heat increase, and so on. Then there's fever and all the ways that fever helps to either kill off pathogens or at the very least make things uncomfortable for the pathogens. There's the interferons that are involved in, especially around preventing or at least blocking the effects of virus pathogens. And then the complement system that allows for opsonization, attracting phagocytes, and stimulating inflammation, and then causing cells to lyse. So, that's a lot in there, but I have confidence in you. So, am I going to ask you to trace the complement pathways? Rest assured, no, I'm not going to ask you to do that. But I do want you to understand how opsonization, triggering inflammation, and those MAC proteins, how that occurs, and how those acute phase proteins do their work. I won't ask you to know what complement protein is the next in that pathway, though. So I'm not going to ask you to get down to that level of detail. All right, you know what to do. If you have any questions, reach out to me and we can talk.